Good again.
It is recommended to wear appropriate personal protective equipment, gloves, surgical mask, eye protection, when collecting samples. Wash hands. Put on gloves and surgical mask. Have the patient facing a strong light to ensure the area to be swabbed is visible. Open the package containing the hydroflox swab and aseptically remove from package. Have the patient tilt their head backwards, open their mouth, and stick out their tongue. Use the sterile hydroflox swab to swab both tonsillar arches and the posterior nasopharynx without touching the sides of the mouth. Break the swab handle at scored breakpoint line. Replace cap and tighten to secure. Remove and dispose of gloves and mask properly. Label vial with appropriate patient information.
Okay. Throat swab specimen.
Deliver samples to laboratory for testing.
Okay. Okay, now it is your turn. Okay, let's go ahead and send your answers. A colony, okay. How we describe colony? It should be as a colony, as a okay. Go ahead and send your answers. Two more. Okay, a growth of bacteria that result from a single bacterium. A colony, A is the first answer, okay? A growth of bacteria that result from a single bacterium, okay? Okay, the bacterial growth on artificial media or in culture typically requires which period of the time? Okay, 24, 48 hours is correct. Good. Media that contain dyes, antibiotics, or other chemical compounds that inhibit certain bacteria while allowing others to grow are called enrichment, differential, supportive, selective. Okay. Selective media is the correct. It selected one type of the microorganisms, okay, and doesn't allow others to grow. Good. Okay. Now let's go ahead, and I have some other questions for you guys. Okay, genitourinary cultures, the third common culture type of cultures that we performing in the clinical microbiology. Okay, it's how the way that we culturing is based on the agents that cause infections. For chlamydia, we have different type of culture medium. for gonococcal organisms different because they like to grow in a different environments. And okay, culture and molecular diagnostic testing, two most common techniques that we are using for diagnosis of genitourinary infections. Now, as you see here, the Jembeck plate for isolation of Neisseria gonorrhoeae, the most common urinary infection is. This system include modified Trier-Martin medium in a synaptop bags and carbon dioxide generating sodium bicarbonate pellet, which is in black color here. After inoculation, the medium is placed into a zip locking plastic envelope and send it to the laboratory. Okay, blood culture. Okay, maybe most important. Okay, and type of the culture in the microbiology. We know normally the blood is sterile. Okay, a single microorganism in the blood is serious. Okay, bacteria can be detected in a blood in the absence of the disease, is known a bacteremia. You may find bacteria in a blood without any infection disease, is known bacteremia. Okay, bacteremia can have a serious consequences for the patients, and blood culture can provide a clinical diagnosis of bacteremia. Okay, now septicemia or sepsis indicated a situation in which the bacteria are actively multiplying and metabolizing, producing toxins and causing harm to the host, to the patients. Okay, which is most worst case scenario. Okay, which is most serious conditions. Okay, septicemia, because it's life-threatening. You have to identify the cause of the septicemia as soon as possible and start with treatment. Okay, now you see here the portal of entry for organism that can cause bacteremia or septicemia. The number one is the genitourinary tracts, especially in females. In females with the immune system compromised patients. And second is respiratory tracts, abscess, surgical wound infection, biliary tract, and others also are portals of entry for organisms. Okay, the most common cause of the septicemia, the most common microorganism that we see in the blood culture, as you see here, gram-positive cocci, such as Staphylococcus aureus, coagulase negative staphylococci, the viridans streptococci, and Streptococcus pneumonia or Enterococcus. These are a most common agents that cause a septicemia, sepsis, but I mean it depends on the age, or genders. Let's see the the and okay, it's based on the ages that what kind of these agents that we see most commonly. Okay, the most common negative gram-negative organisms that we can found in the blood are E. coli, Klebsiella pneumonia, Pseudomonas aeruginosa, and Enterobacter, okay, and Proteus. Okay, let's see Proteus that we mostly see in neonates, in babies, newborn babies. Okay, however, Klebsiella pneumonia or Pseudomonas aeruginosa and Enterobacter in older patients. Okay, a progressive syndrome, which is non-septic shock, can occur in the blood because of release of the endotoxin and a part of the cell wall of the gram-negative bacteria. This kind of sepsis is really serious, life-threatening infectious disease. Okay, because they produce endotoxins, okay, which is result in septic shock. Okay, obtaining a sample for blood culture is really important. We have to pay attention, be carefully. First, we have to cleansing the collecting sites as we are performing a surgical operation, okay, via alcohol or some kind of iodine solution, and then allow them to dry for 1 minute, and then take withdraw a blood. Now I am going to skip these videos. This is really good videos how we obtain a sample for blood culture. Okay, there are different type of the culture media for blood, as you see here, for aerobe microorganism, for anaerobe. Here is medium for, and also the medium for pediatric patients is the content of these mediums may be differ from each other, okay, in order to let the microorganism grow enough. Okay, now the last step of the bacterial identification is the antimicrobial susceptibility test, or antibiogram. It is important, okay, to do that because by doing this, you provide a information to the physicians, okay, to choose a appropriate antibiotics, and then to treat the disease effectively and efficiently. Okay. Okay, the laboratory report showing susceptibility or resistance, okay, to the particular antibiotic largely determine whether the agent is used or withdrawn. Now, if you diagnose your patients, okay, you withdraw the samples, you obtain the samples before you started with antibiotics, okay? You start with a kind of antibiotic that you think that it is effective, okay? However, At the same time, before you started with antibiotics, you collected a specimens, okay, sample, sent to the laboratory for antibiograms. Okay, once you receive the result of the antibiograms, and you will see whether your treatment is appropriate or not. Okay, and now a result of susceptible indicate that the patients most likely respond to the treatment with the antimicrobial agents. A result of resistance indicate the treatment with the agent will the most likely fail. Okay, it doesn't kill the bacteria. Okay, therefore we are reporting as a susceptible or resistance. Okay, now I'm going to show you in a few slides. Okay, there are here two definitions that we need to know. One is the minimal inhibitory concentration and minimal bactericidal concentration. Okay, the lowest concentration of the antimicrobial agents, antibiotics, that inhibit the growth of the organism is known minimal inhibitory concentration. Okay, and the lowest concentration of the antimicrobial agents that will kill the microorganism is known minimum bactericidal concentration. These two definitions are most common definition that we are using in terms of antimicrobial susceptibility test. Okay, now you see here the agar diffusion, the antibiograms, how look like. Okay, this is okay, here the a kind of the mediums which we call Miller-Hinton agar has been inoculated with a test or with the test organism and antibiotic disc, this white oval shape are antibiotic discs have been placed into the media, okay, and then okay, there is no incubation has yet occurred here, and then you okay, this media also they contain a kind of the microorganisms, okay, and then you place the antibiotic disc on the media, and then you incubate this place in incubators for 16 to 80 hours. After that, as you see here now, each disc represent a different kind of antimicrobial agents, antibiotics. Okay, and they also we know which these represent which kind of antibiotics. Okay, now you see here, this is a zone of inhibition. What does mean? If the more the zone of the inhibition is higher, that means these antibiotics is more effective. Okay, the zone of inhibition means the antibiotics, like antimicrobial agents, doesn't allow the microorganisms to grow. It kills the bacteria. Okay, here this is susceptible to these antibiotics. Okay, but here is resistance to these antibiotics. Okay, because there is no zone of inhibition here. The microorganisms are growing here around these antibiotics. Okay, this is important to know whether your antibiotics is susceptible okay, that the microorganism is susceptible to the antibiotics that you are using. Okay. Okay, now let's talk about different kind of the microorganisms and start with the microbacteria. Microbacteria are acid phase gram-positive bacilli, okay, are obligate aerobic microorganisms. There are three main groups of these type of bacteria. Mycobacteria tuberculosis, as the name implies, the cause of the tuberculosis, may be most common type of the lung infection disease that caused by mycobacteria. There are other type of the mycobacteria, such as Mycobacteria lepra or non-tuberculous mycobacteria. And there are, okay, we are using sputum as a specimen to identify, to diagnose tuberculosis. And we cultivating this specimen in a different kind of the culture medium, such as Levenstein, Janssen medium, okay? And we are using acid fast stain as a techniques to identify Mycobacteria tuberculosis, and acid fast stain using Kenyan carboxy methods is other type of the techniques that we are using to identify Mycobacteria tuberculosis. Okay, fungi, okay, issues of the mycology, okay, are yeast or molds, okay. Fungi are eukaryotes, right? And they possessing a true nucleus with a nuclear membrane, such as cell of the humans or animals. And fungal cell wall are not composite of the peptidoglycan, such as bacterial, but they contain alternate carbohydrates such as chitin. Okay, yeast are single celled organisms that produce budding, okay, and molds have a basic structure that consists of a tube-like filamentous projections are called hyphia. Now I am going to show you hyphia here, okay? A pseudohyphia consisting of elongated cells with constrictions of attachment sites here. Okay, fungi are as a source of infections. There are many fungi, some of them a cause of infections. We are interested in a fungi which are a cause of infection in human beings. Okay, collection of specimen for fungal studies are important, and you cannot let them grow in all the mediums. There are a special medium for fungi, okay, and the methods for detection are different. Direct microscopic examination of fungi is most common one, and easiest and also cheaper techniques that we are using to identify fungi. Gram stain techniques, okay, potassium hydroxide preparation, India ink, acid fast stain, that are some techniques that we are using to identify fungi via direct microscopic examination. as I said, these are most common techniques and really easier to do to perform that. Okay, as you see, gram stain of the yeast here. Okay. Yeast are not classified as a gram-positive because of absorption of the gentian violet stains. However, we are using gram stains to identify them. Okay, and an example of India ink stain of Cryptococcus neoformans, maybe one of the most common cause of fungi disease in the United States, in some areas. Okay. As I said, the culture media for fungi is special. Okay. There are here examination of the culture. Germ trip test is a technique that we're using to identify Candida albicans. Okay, as you see, the Candida albicans grow faster at the 35 Celsius degree. How it looks like, the germ tube test for a Candida albicans in a sheep or rabbit serums is a distinct appearance. If you see that, you will be able to diagnose, to identify it. Okay, parasitology is a main department of the microbiology. You know, there are many parasites who they are living in or on their host at the expense of the host, and some parasites cannot survive without their designated host. Other parasites can exist in a free living state as an intermediate state before transmission to the suitable host. And we also know there are some beneficial parasites, they living in our gastrointestinal tracts, or which with no harm coming to the host. The relationship is beneficial to both, and which we call symbiotic life. Okay, parasite as a source of infection is important for us that since we are living here in the United States because of the great influx of the refugee population. Okay, that means many organisms endemic elsewhere are being identified in the United States. Okay, how we identify them? Okay, the stool specimen is important. In some cases, we also using blood specimen to identify them. Okay, the methods for detection of parasites, wet mount or direct smear, these are also most common techniques that we are in parasitology. And I am going to show you here the Trichomonas vaginalis that can inhabit the urogenital system of both males and females. It's considered a pathogenic parasites. The Trichomonas vaginalis is the cause of vaginitis, urethritis, and prostatitis. Okay, as you see here, the trophozoite form of Trichomonas vaginalis, how look like. Okay, there are different techniques that we are using to identify Trichomonas vaginalis. Rapid test for identifying its, as is here, the self-contained system for culturing Trichomonas vaginalis. Okay. As I said, intestinal ova or parasites under microscope, or under which we call direct smear, is most common one, and microscopic examination of the feces of the stool, microscopic examination. Sometimes you need to concentrate the sample in order to be able to see the microorganisms, the parasites, permanent stain of the smear, molecular diagnostic, and direct antigen detection also are techniques that we are using to identify the cause of the parasitic infections. Now, as you see here, maybe the most common parasitic infections, which is which causes by Enterobius vermicularis or pinworms, most common parasite in children worldwide. Okay, and it is a roundworm, okay, whose adult female migrates during the night and depositing her eggs in the perianal region. This is egg of the pinworms. Okay, now how we can identify it? The technique that we are using to identify pinworms is known as cellophane tape collection methods. Okay, a piece of clear cellophane tape with the sticky side toward to the patient is pressed against the skin across the anal opening to collect any eggs using even through pressure. And then this sticky side of the tape is then placed down against the surface of a clean glass slide with a glass cover slip, and then you are going to examine under microscope and you see the eggs. Okay, the eggs that you see with cellophane tape preparation. Okay, tick-borne disease. Okay, in some areas they are common. Okay, I was living in Minnesota. I mean, most common parasitic infections that we encounter in the Minnesota. Okay, in some area in the U.S., you will see. Okay, and tick-borne infection agents can cause babiosis, Lyme disease, ehrlichiosis. Okay, these are three diseases caused by tick-borne. Okay, malaria, the maybe most common disease in the Africa, but you also may see in the US, and Plasmodium malaria, the cause of the malarial disease. It's placed in the red blood cells. Okay, the malarial parasites enter the human through the bite from the infected Anopheles mosquito, and then the malarial parasites enter in the bloodstream and invade red blood cells, and then will damage these red blood cells later. Okay, the tin or tick smear are prepared, stained with Wright or Giemsa. These are the kind of the staining techniques, and then examine under microscope. And as you see here, these are infected by Plasmodium malaria, the red blood cells. Okay, virology. Okay, still we have some times. Okay, viruses cannot replicate outside of the another of the living host cells. The virus relies on host cells machinery to reproduce the viral particles for propagation, transmission, and infections. Virus particles are referred to as virus and consist of three major parts. These are nucleic acid core, capsid, and envelope. Okay, but they need to live on a host in order to use a DNA of the host to grow, okay, to replicate. Okay, there are, we know viruses as a source of infections, which we are interested in, and the collection of specimens for viral infections is different from bacterial infections. There are few cultures that we're using for viruses because it is really hard to let them grow in the culture. And therefore, the most common techniques that we're using to identify viruses are immunodiagnostic techniques and molecular diagnostic tests. The antigen-antibody detections is the most common methods that we identify viruses. Now, point-of-care testings that we already discussed before. There are rapid testing for streptococcal infections, for pneumococcus and Legionella, for HIV, IV, and also there is a rapid test for Plasmodium falciparum, okay, which is other cause of the malaria, okay. There are Plasmodium falciparum, Plasmodium malaria, and Plasmodium vivax, three common cause of the malaria. Okay, automation in microbiology, the last part of the microbiology that I want to share with you. We like automation, right? Because it reduces the rate of errors. Okay, it make it decrease the test around time. Okay, you will provide the test in a short time, and then, as you see here, there are a few automation systems that we nowadays using in the microbiology. Mass spectrophotometry, a MALDI-TOF, has standardized microbe identification after only 6 to 9 hours. Okay, with the traditional methods, we need 3 days, 5 days to identify microorganisms. But with the MALDI-TOF, you will be able to identify microbes in a 6 or 9 hours. Okay, continuous monitoring blood culture systems are other automation systems. Automated microbial identification, automated antimicrobial susceptibility testing systems, antibiogram systems are widely used in microbiology laboratories. These are the most common automated system that we are using in clinical microbiology. Okay, now with this, questions, I am going to finish these lectures. And let's see now. And go ahead and send your answers. Come on, guys. Okay, acid-fast gram-positive bacilli is mycobacteria. Okay, mycobacteria tuberculosis is a cause of tuberculosis, right? It is important, okay, maybe one of the common diseases that we see in the United States in terms of mycobacteria. Okay, the common parasites identified in the urogenital system is Cryptococcus neoformans, Trichomonas vaginalis, Enterobius vermicularis, Candida albicans. The most common parasites identified in the urogenital system. Okay, Trichomonas vaginalis is the correct one. Urogenital system, okay. Then, okay, you can see both in female and male, okay, but this is the most common parasite that we identify in urogenital area. Okay, now which of following microorganisms enter the human through the bite from an infected Anopheles mosquito? Plasmodium malaria, correct. Good job, guys. Okay, now, can you see now?
It looks the same.
Okay, okay. Now the correct answer, okay, let's see this. The correct answer is B. As you see, the inhibition zone. Can you see the inhibition zone? Okay, this, as I said, all these represent one antibiotics, one antimicrobial agents, and these are, okay, these antibiotics, okay, the microorganisms is susceptible to these antibiotics, which have inhibition zone, okay, and resistance to these antibiotics, okay. Now let's go ahead. Okay, now, can you see my screen?
Yes, we can.
Okay, why does this differ from other types of microorganisms? Because they cannot replicate outside of another living host or rely on host cell machinery to produce the viral particles for propagation and so on. Okay, send your answers now. Oh, okay, let's see. Okay. Both A and B is correct. Okay. The correct answer is D. Okay, last one. The depicted microscopic picture represents which of the following microorganisms? Tick-borne, Plasmodium malaria, Anthrabius vermicularis, Trichomonas vaginalis, Candida albicans. The egg of which of the following? Okay, let's see. Okay, none of that. Anthrabius vermicularis. Okay. The correct answer is C. Okay, no problem. We are going to learn them. Okay. Okay, that should be enough for today.